All right. So we're going to talk a little bit now about lipid metabolism. This is the diagram for it. Again, I just put these here for you guys. I'll give you the slides after the lecture. And you can always come back and take a look, but I think I also put these on all of the slides after. Maybe not. Okay, beta oxidation is the first part of lipid metabolism we are going to talk about. So after being absorbed by the digestive tract, triacylglycerols, some of you might know them as triglycerides, are transported through the blood and lymphatic system to the organs in little lipoprotein vesicles known as chylomicrons. So those transport your fat throughout the body because obviously they are hydrophobic. Fatty acids, fat molecules are hydrophobic, so they can't travel through blood and lymph without some sort of vesicle or some sort of carrier protein. So that's what chylomicrons do. And once they get to the target tissue, the triacylglycerol is broken down into 3 fatty acids and then activated before it crosses the mitochondrial membrane. So when I say activated, I mean that acyl-CoA synthetase converts a fatty acid into acyl-CoA by hydrolyzing ATP to form AMP, as we were talking about before, and a pyrophosphate. So the pyrophosphate are those 2 joined phosphate groups. And acyl-CoA, that ACYL, is sort of just a generic term that we're talking about because we're not really, that would change depending on how many carbons are in the fatty acid chain. So for example, if we had 16, it would be palmitoyl-CoA, acetyl-CoA is 2. So acyl is just a generic name for the fatty acid chain that's connected to the CoA. So that's what that means when we say acyl-CoA. So when an acyl-CoA is in the mitochondrial matrix, a series of 4 reactions repeats on the chain molecule until the entire chain has been converted into acetyl-CoA molecules. So it's sort of like the Krebs cycle as well, where we're kind of just repeating. It's not a true cycle because different molecules come in, but we're just doing these 4 reactions repeated and repeated and repeated until we're only left with acetyl-CoA molecules. So the starting product of beta oxidation is going to be these triacylglycerols, and the end product is going to be a bunch of acetyl-CoA. So long-chain fatty acids must enter the mitochondrion using the carnitine shuttle. So short-chain fatty acids can just diffuse through the membrane. It's hydrophobic, but if they're small enough, they can just diffuse through. It's not going to be too much of a problem. But a lot of fatty acids are longer chains, and they do have to use this, this thing that we call the carnitine shuttle. So basically all it is we're just attaching it to a molecule, we're throwing it through the membrane, and then we're detaching the molecule and sending that molecule back outside. So is the first step. Carnitine palmitoyl transferase 1 transfers the acyl-CoA to the hydroxyl group of the carnitine. So we have a carnitine molecule, we're taking the acyl-CoA molecule that we really want, that we're interested in, we're just attaching it to carnitine and specifically on the hydroxyl group. So step 2, the new acyl carnitine is what we're referring to this molecule as, is transported into the mitochondrion via the transport protein carnitine acyl carnitine translocase. So this transport protein is an integral protein. It's in the membrane, and the acyl-CoA cannot go through this translocase unless it's attached to a carnitine. So that's why we're attaching it. It's going to go through this translocase. Simultaneously, an unpaired carnitine molecule is transported out of the mitochondria. So it's what we call a symporter or an antiporter, I'm sorry. One is going in and one is going out. So the carnitine, the acyl carnitine is going in because we want to get that acyl group in, and then just the unpaired carnitine molecule is going out because it's probably going to be used for another shuttle later. So that's sort of how we maintain that equilibrium of things going in and out. So once the acyl carnitine is inside the mitochondrion, carnitine palmitoyl transferase 2 converts the acyl carnitine back into acyl-CoA and carnitine once it's inside the mitochondrion. So it's the palmitoyl transferase 2 does the exact opposite as 1. It just rips them apart. And then the carnitine is transported back out of the matrix, like what we just said in step 2, and the process just repeats, and that's how we get these fatty acids inside. So we're just attaching it to a carnitine, putting it inside for the translocase, ripping them apart, and sending the carnitine back outside. That's all it is. And then, like I said, the smaller chain fatty acids can diffuse through the membrane. Okay, so beta oxidation is actually a series of four reactions as before. They act upon the fatty acid chain, cutting off two carbons from the end to form acetyl-CoA for each cycle. So we have a bunch, the fatty acids, it's just carbons, it's just a carbon chain, that's all a fatty acid is. And we're cutting off from the end. That's what beta oxidation is. So acetyl-CoA is just a two carbon molecule. So we're basically just modifying and cutting off these two carbons until we finally get down to only two carbons, and we just call it an acetyl-CoA, and the entire molecule has been separated into two carbon acetyl-CoAs. And it happens in 4 steps. So in the first step, acyl-CoA dehydrogenase is the name of the enzyme, and it's used to remove a hydrogen and form a trans double bond between C2 and C3. So let me see, acyl-CoA dehydrogenase. So we have this fatty acyl-CoA right over here, right up at the top on this diagram, if you guys can see. It's coming in. So we have this acyl-CoA dehydrogenase, and you can see the blue and red carbons. Those are the alpha and beta carbons that we call them. Once it's finished with step 1, we have this double bond right over here, and it's going to be trans because these two hydrogens are on the opposite sides. And we also create an FADH2 in the process. So that's good. So enoyl-CoA hydratase then hydrates the double bond and adds a hydroxyl group to C3. So that's the second step. If we look right over here, we take this molecule that we just created, the enoyl-CoA hydratase, adds H2O. So we look right over here at the beta carbon, this blue beta carbon. We now have a hydroxyl group and a hydrogen, as opposed to when we look back up before, we just had this double bond. So this was not saturated. We now have a completely saturated carbon over here. The hydrogen was also added back to the alpha carbon and added back to the other side of the beta. So we have a saturated molecule with a hydroxyl group now. And that's on C3. That's important to know. So beta oxidation, read that already. So the hydroxyl group in step 3 becomes a carbonyl in another dehydration reaction catalyzed by 3-hydroxyacyl-CoA dehydrogenase. So we can look right over here. It's going to be another dehydration reaction. So we're going to be making, again, another carbonyl group. We're going to have a double bonded oxygen. So we go from this step over here. We're creating another NADH, one of those high energy electron carriers. And we now, on the beta carbon, have a double bonded oxygen. And like I said, NADH is produced. So this next step is thiolysis, as I was talking about before. It's facilitated by thiolase. The bond between C2 and C3 is thiolized, and a CoA group attacks the C3. To reform acyl-CoA, and that acyl-CoA is now going to be 2 carbons shorter than the original because we just let go of an acetyl-CoA. So we look over here. It's going to be broken right between this blue and this red, right between this alpha and this beta. It's going to split off. This R group just means it's the rest of the acid. So this left part is actually going to be the rest of the 16 carbon chain, say, if we had a 16 carbon molecule. So this all over here is going to remain. And these two, the alpha and everything to the right, is going to attach, it's going to separate. And this CoA molecule, it's going to become acetyl-CoA over here. So we move up and we see that this carbon, this beta carbon that's blue, is now attached to a sulfur and another CoA. So we added another coenzyme A group on, but now since these two are gone, it's no longer a 16 carbon, it's now a 14 carbon. So that's basically what beta oxidation is. It's just those 4 steps being repeated. So it's going to repeat again as a 14 chain carbon. It's going to do the same thing. We're going to come right over here. We are going to dehydrate, excuse me. We're going to rehydrate with a hydroxyl group over here. We're going to dehydrate again, and then we're going to thiolize. We're now going to have a 12 carbon group. We're going to do the same exact thing. Another acetyl-CoA is going to be released, and we're going to have a 10. And then it just keeps on going and going and going and going until we finally have 4 carbons. We split them in half, and both of them become acetyl-CoA. And we just made a bunch of acetyl-CoA from a fatty acid, and that is how beta oxidation works. And then the acetyl-CoA can, move to other parts of the body, and it can be used to spin the Krebs cycle. So 20% of you said B and 80% of you said C. So the correct answer actually is B. And I'm glad I finally stumped you guys because I felt like my questions were too easy. But we'll go over it. So how many rounds of beta oxidation would palmitic acid, which has 16 carbons, undergo before it's completely degraded? So if we think about it, we have 16 carbons. We cut 2 off. We now have 14. That's 1 round. 14, we cut another one off. We now have 12 carbons, 2 round. We cut another one off, 10 carbons, 3. 8 carbons, 4. 6 carbons, 5. 4 carbons, 6. And then we're going to do 1 more cut between those 4 carbons for 2 acetyl groups, which makes 7. So that's a pretty good thing to know, is that anytime you're doing beta oxidation, it's going to be 1 less than half of the amount of carbons. Just because that last one, when we cut, we don't need to cut anymore because that acetyl-CoA is just going to be there. So the formula, like the formal formula, would be N divided by 2 minus 1. So we have 16, where N is the number of carbons on the fatty acid. So we have 16 carbons. 16 divided by 2 is 8. You might normally think that it's going to be 8 rounds of beta oxidation, but you have to minus 1 because that 7th oxidation, what's remaining is just going to be 2 carbons. So we don't need to do another round of beta oxidation. We just have to tack a coenzyme A on there, and we have our acetyl-CoA. Okay, so we talked about oxidation, which was the catabolic process. So now we're going to talk about fatty acid synthesis really quickly. So catabolism takes place in the cytoplasm. The oxidation is a form of catabolism. It's fatty acid catabolism. But oxidation, the anabolism synthesis, takes place in the mitochondrial matrix. I think that might be. Anabolism is not oxidation. Catabolism is oxidation. So I'm going to fix that. Yeah, but we're talking about anabolism now. So anabolism is fatty acid synthesis, and it's going to take place in the mitochondrial matrix. So the first and committed step of fatty acid synthesis is the conversion of acetyl-CoA to malonyl-CoA, and it's catalyzed by acetyl-CoA carboxylase. So the first step of fatty acid synthesis is its committed step. So once that happens, it's turning into a fatty acid, whether it likes it or not. And that's right over here. So we have the acetyl-CoA. CO2 is actually taken in. ATP is hydrolyzed into ADP, and it also uses biotin. Acetyl-CoA carboxylase. I believe biotin is the cofactor for acetyl-CoA carboxylase. And all we're really doing, all we're really doing is adding this right over here, just this phosphoryl group right over here. We're just adding it to acetyl-CoA, and that becomes malonyl-CoA. So that's the first step of fatty acid synthesis. So does everyone see that? It's sort of like the opposite of decarboxylation. We've sort of been going the other way. We've been taking these carboxyl groups and taking them off as a CO2. Now we're taking in the CO2 and we're adding a carboxyl group and creating malonyl-CoA. Okay, so fatty acid synthase is a very large enzyme that has multiple catalytic domains. The binding domain is known as the acyl carrier protein. Acetyl-CoA binds to the ACP first. So that's sort of like the first domain. It's the binding domain. So we have a molecule. Remember, we made the malonyl-CoA, but we also still have acetyl-CoAs that are just floating around. So acetyl-CoA is going to bind to the ACP. And then it's going to be shifted over to another domain and held in place by a cysteine residue. So remember, the acetyl-CoA, coenzyme A, has that thiol group. It has that sulfur group. So it can react with cysteine residues. It's going to make a disulfide bridge, and it's going to hold it in place pretty well. So it binds to the ACP, and then they shift it over to another domain that has a cysteine. So it's sort of like shifting it over into a little, like a holding place. So it's going to hold that acetyl-CoA. Then we have malonyl-CoA that's going to bind to the ACP, that binding domain. So we now have malonyl-CoA and acetyl-CoA right next to each other. So we're going to condense them together And in that condensation reaction, the carboxyl group from malonyl-CoA is going to decarboxylate and be released as a carbon dioxide. That's where the energy of the reaction comes from. And the 4-carbon chain is then reduced twice by NADPH. We've been talking about NADH, but NADPH is also a high-energy electron carrier. It's created in the phosphate pentose pathway, pentose phosphate. And it shifts over to the cysteine domain. The 4-carbon chain is reduced, shifts over to the cysteine domain, and then it just repeats the process again. So another malonyl-CoA enters the ACP, and the process restarts. We're just condensing, decarboxylating, and then shifting over to the cysteine domain to hold it. So it basically is just beta oxidation in reverse. And this is the schematic that I put to the left. ACPCO2, we're removing carbon dioxide. We're going to condense the acetyl-CoA and the malonyl-CoA. And then we have these two reductions with the NADPH. So it's going to reduce. There's another dehydration reaction in between. It's going to reduce. And then this is our product. We are now 2 carbons longer for our fatty acid. And the same thing, it feeds right back in. So it's just a cycle. As you can see, a lot of metabolism is just cycle, just because it's super efficient. And the process repeats until a 16-carbon fatty acid is produced. So it always goes up to 16 carbons. It produces palmitic acid. That's your finishing fatty acid. We're done. Okay, we're going to talk really quickly about ketogenesis. So ketogenesis occurs during periods of starvation when blood glucose and glycogen stores have been depleted. So obviously, carbohydrates are the body's preferred source. Glucose is the body's preferred source of energy. So when we can't get it from the blood, we look to glycogen. When we can't get it to glycogen, we got to look somewhere else. So there are a couple of other things that'll happen. You know, our body will start gluconeogenesis, but ketogenesis also increases because we don't have any fuel, basically. So when this happens, the mitochondrial matrix produces what we call ketone bodies, and the process in which they are created is known as ketogenesis. So ketone bodies, the really important thing about them is that they are able to cross the blood-brain barrier, and they can provide the brain with energy when glucose is absent. So that's really important. The brain loves glucose. It is definitely its preferred source of energy, but when there's no glucose present, there's really nothing you can do. So the body switches over to make ketone bodies, and it tries to get them right up to the brain. So they are transported into the brain, and it's converted back into acetyl-CoA before entering the Krebs cycle. So we don't even need to do glycolysis because obviously there's no glucose. We make the ketone bodies, it gets to where it needs to be. It's broken down into acetyl-CoA, and we can start spinning the Krebs cycle again. We can start making NADH, FADH2, and we can move those over to the electron transport chain to create some ATP. The final products are 3 ketone bodies. It creates 3. Beta-hydroxybutyrate, acetoacetate, and acetone. It actually first creates acetoacetate, and then the acetoacetate can be transformed into either beta-hydroxybutyrate or acetone. Beta-hydroxybutyrate is actually the most common ketone body that you'll see throughout, and acetone is the least common. But acetone is actually responsible for when diabetics go into ketoacidosis and you actually can smell like acetone on their breath. It's just because they're in such a state of ketoacidosis that even though acetone is the least common ketone body, its presence in the blood is so high that we're actually breathing it out because it's very volatile, so it's going to come out in your breath. Ketone bodies are negative, so that's why it contributes to ketoacidosis. It lowers the pH in the body, and then all of our compensatory mechanisms are going to start. We'll hyperventilate. We're going to breathe really fast because we're trying to get the carbon dioxide out of our body to increase our pH. Our heart rate is going to increase, all of that stuff. So ketogenesis is regulated by the availability of carbohydrate energy sources in the body and how it affects the acetyl-CoA levels. So the carbohydrate availability will affect acetyl-CoA levels, and the acetyl-CoA levels are really what's going to affect the ketogenesis. So when the body does not have free carbohydrate fuel, fatty acid oxidation creates an abundance of acetyl-CoA to compensate. So that beta oxidation is going to be creating a bunch of acetyl-CoA because there's no there's no free carbohydrates. So beta oxidation is just going to be going because it's trying to make that energy. Under normal conditions, the acetyl-CoA would feed into the Krebs cycle to produce higher energy precursors for the ETC. However, when ketogenesis is occurring, it's going to be because of starvation conditions. So under these starvation conditions, the intermediates from the Krebs cycle, mainly oxaloacetate, they're going to be depleted because of the activation of gluconeogenesis. So a lot of the intermediates in the Krebs cycle are also precursors and intermediates in gluconeogenesis. So when we're under starvation conditions, we're going to shift our resources from the Krebs cycle to gluconeogenesis because we really need to make glucose. We have to get our blood glucose up. So that's going to take priority over the Krebs cycle. And because that's happening, even though we have this abundance of acetyl-CoA now from this oxidation, we're not going to be able to spin the Krebs cycle as efficiently because we don't have everything we need. It's all going to gluconeogenesis. So this is actually driving the activity of the Krebs cycle down. It's down regulating the Krebs cycle. But this high level acetyl-CoA is actually what activates ketogenesis. So those acetyl-CoAs are going to be turned into ketone bodies. So we had 20% of you say A, nobody said B, 80% of you said C, and nobody said D, and nobody said I don't know, which is cool because the correct answer is C, in fact. So let's look at the question a little more closely. A type 1 diabetic experiencing an episode of ketoacidosis would most likely exhibit all of the following symptoms except, and it's not really super important that you even know the difference between the two types of diabetes, just the fact that diabetes leads to an increase in your blood glucose levels. So they would exhibit all the following symptoms except low intracellular levels of glucose. That would be true because all of their blood is basically sequestered. All of their glucose is basically sequestered in the blood. So whether it's because there's no insulin present or because there's an insulin resistance for the cells, regardless of either type of diabetes, that's not going to be true. That is going to be true. There's going to be low intracellular levels. So B, an increase in acetone and other ketones found in her breath. That's also going to be true. Like we just spoke about that a few minutes ago. Ketoacidosis, when we can't get fuel into our cells, we upregulate ketosis. We go into a state of ketosis, so acetone and other ketones are going to be very highly concentrated throughout our body, and they're going to be very highly concentrated in our breath. D, an increase in ketogenesis would also be true. If it weren't true, then B wouldn't be true because we don't have that fuel. C is incorrect because high levels of glucagon secretion from the pancreas would not occur. Glucagon is secreted when there are low blood glucose levels, and we would not expect a person experiencing ketoacidosis to have low blood glucose levels. They will probably have very high blood glucose levels, and they just can't get the glucose into their cells.